Hi guys, I am Leila Mamagon from Criminology 24 and I am here to discuss with you about the scientific methods of personal identification other than fingerprinting. There are 6 scientific methods: the anthropometry, portrait parley, forensic odontology, forensic anthropology, forensic serology, and the other one is DNA fingerprinting. The first one is anthropometry.
Anthropometry Bertillon system. The science dealing with the measurement of human body to determine differences in individuals. It was introduced by Alfonso Bertillon in 1900 and spread it through the world. In 1914, fingerprints was officially adopted in France, replacing the Bertillon system. Composition of Bertillon system. 1. Anthropometrical signalement, the measurement of the body, head, and limbs. Number 2. Descriptive signalement, a clear and specific system, a description of an individual as well as the photography of the malefactors. Number 3. Peculiar mark signalement, by indelible marks like scars, tattooing, birthmarks, mole, etc. Example of anthropometry. Yves Klein, entitled Anthropometry, 1960. The Radical Nudes of Yves Klein's Anthropometries, 1960.
Anthropometry is the study of human dimensions.
Everything from body fat percentage to the length of your nose can be considered anthropometry.
For this section of testing, we have 4 measures.
First is height, without shoes.
Then arm span and hand span.
The key takeaway from these tests is the ratio between your height and arm span. An arm span longer than your height is usually best, as is a larger hand span.
And last is body weight.
Body weight alone doesn't actually tell you much. Negative things like dehydration or having low glycogen stores can actually reduce your weight, while positive factors like gaining muscle mass, or if you're a junior, just growing taller, is going to have a positive effect on your performance, but your weight's going to go up as a result. What body weight does give you though is a general radar for your health. Smooth, gradual, even fluctuations in body weight are much more conducive of high performance than sharp peaks and troughs.
The second one is portrait parley.
Portrait parley, or speaking likeness in French. Verbal and picturesque description of the physical characteristics of the subject or the person being identified. Such information may be given by witness, relatives, or person who is acquainted with the physical features of the person to be identified. In order to recognize individuals who were repeatedly arrested, Bertillon developed means by which portraits could be sorted by common morphological characteristics, the specific shapes of the different parts of the face, and thus an individual's prior photo could be found without having to resort to browsing through large collections of portraits. This classification is known as the portrait parlé or spoken portrait. Bertillon card. The Bertillon card included 2 photographs, one face-on and one in profile.
The third one is forensic odontology.
Dental identification, forensic odontology, proper collection, interpretation, evaluation, as well as presentation in court of dental evidence during the conduct of criminal and civil proceedings. Odontological identification is based on systemic comparison of pre- and post-mortem dental characteristics of individuals based on dental records and the supporting radiographs. But this technique is complicated by the trauma to just and inadequate ante-mortem dental information. According to American Board of Forensic Odontology, dental identification can be divided into 4 types. 1. Positive identification. The ante-mortem and post-mortem data match to establish that it is from the same individual. 2. Possible identification. The ante-mortem and post-mortem data have few consistent features, but because of quality of the records, it is difficult to establish the identity. And number 3, insufficient evidence. The data is not enough to form the conclusion. And number 4, exclusion. The ante-mortem and post-mortem data clearly inconsistent.
The fourth one is the forensic anthropology.
Forensic anthropology can be described as the analysis of human remains for the medico-legal purpose of establishing identity. Anthropologists are routinely called upon to assist the identification of individuals whose remains are severely decomposed, burned, disrupted, mutilated, or otherwise rendered difficult to recognize. Their services have been of particular value in 3 main areas. 1. Investigations resulting from homicide, unexplained natural deaths, accidents, and mass fatality. 2. Non-criminal events resulting in multiple deaths, example, arising from natural catastrophes. 3. War crimes investigations and genocide. Forensic anthropology and human identification. The role of the forensic anthropologist focuses on applying detailed knowledge of the development, morphology, and variation of the human body as an aid to establishing, confirming, or indeed refuting personal identification. 4 basic biological criteria form the basis of any primary identification. 1. Skeletal or developmental age. 2. Biological sex. 3. Living stature. 4. Ancestry or racial affiliation. These characteristics are supplemented by secondary traits which include the following. Number 1. Body modifications. 2. Evidence of surgical interventions. 3. Including scars and prosthetics, evidence trauma. 4. Chronic hard and soft tissue diseases that may leave characteristics macroscopic and microscopic lesions on surviving tissue. Examples of the stages of forensic human identification. Is it human? Differentiating human from non-human bone. Frequently, bone is presented for identification from the sites of accidental discovery, or from the sites to which a presumption of violent or wrongful death may be attached through police intelligence. From a medico-legal standpoint, initially it is presumed that the remains are human and were deposited within a relevant time scale.
The fifth one is the forensic serology.
Forensic serology is the application of the study of blood, semen, saliva, and other body fluids to legal matters. The field generally is comprised of the detection of enzymes and antigen, as in the identification of seminal stains or blood typing. The serology section of a forensic laboratory may deal with any or all of the following: blood typing, characterization of unknown stains, blood spatter analysis for crime reconstruction, paternity testing, semen identification in sexual assault cases, DNA typing for identification of victims and suspects. Blood typing, unknown stains, blood spatter, paternity testing, seminal stain, and DNA typing.
Blood. 10 pints of life force, coursing through thousands of miles of arteries, veins, and capillaries. Those blood vessels carry blood to every other organ, keeping them functioning and thriving. Our red blood cells carry oxygen and nutrients. Our platelets stop bleeding and help to heal our wounds. Our white cells protect us against infection. And the yellow liquid plasma in which they are suspended carries an array of proteins that regulate bleeding.
Castle-Meyer. Solution of phenolphthalein is applied to suspected blood stain, followed by hydrogen peroxide. A bright pink color indicates the presence of blood. Eucomalachite green. Same procedure as Castle-Meyer. Produces blue-green color in the presence of blood. Luminol. Used for large areas where blood is suspected but not visible. If blood is present, it will glow. Must be viewed in darkness and photographed quickly. Once the presumptive test indicates that a stain may be blood, the serologist must confirm that it is a human blood. Precipitin test identifies the presence of proteins that are found only in human blood. How serology helps in crime. Serology allows the forensic scientist to segregate these bodily fluids when found at the scene of the crime, and then perform a variety of tests on them in order to identify where these fluids originated from, or most importantly, who they came from.
Go. And the last one is the DNA fingerprinting.
DNA fingerprinting. DNA profiling is the process of determining an individual's DNA characteristics called a DNA profile. that is very likely to be different in unrelated individuals, thereby being as unique to individuals. The modern process of DNA profiling was developed in 1984 by Sir Alex Jeffreys, while working in the Department of Genetics at the University of Leicester. DNA profiling is most commonly used as a forensic technique in criminal investigations to identify an unidentified person or whose identity needs to be confirmed, or to place a person at a crime scene, or to eliminate a person from consideration. DNA profiling has also been used to help clarify paternity, in immigration disputes, in parentage testing, and in genealogical research or medical research. DNA fingerprinting has also been used in the study of animal and floral population, and in the fields of zoology, botany, and agriculture.
My house was broken into last week. The police can identify the culprit from blood found at the scene. How can they do that? To understand this, we need to state what is meant by DNA fingerprinting and describe some uses of DNA fingerprinting. The burglar smashed a window, leaving blood on the broken glass. Samples of blood, hair or skin can be collected from a crime scene. Forensic scientists investigate the samples in a lab. First, they extract DNA from the cells. They use enzymes to cut the DNA into shorter fragments. The fragments are placed into a well in a gel. They are separated using an electric current. Different sized fragments travel different distances in the gel. The smallest fragments travel the furthest. A pattern of bands is produced. This can be compared with the patterns of DNA from possible suspects. The enzymes cut different people's DNA in different places, producing a different set of fragments. The different sets of DNA fragments will produce different patterns of bands when separated on a gel. Each person has unique DNA, which will produce a unique pattern that can be used to identify them. If two patterns match, the samples are from the same person. So, the police are able to identify the man who burgled my house using his unique DNA fingerprint. They make a DNA fingerprint by taking a sample of cells from someone and separating fragments of DNA to make a pattern. If the pattern matches a sample from a crime scene, that person was there.
That's all for today, guys. I hope you learned something about my discussion about the scientific methods. Bye!